so now I'm in conversation with Steve Gardner, who's the CEO and founder of Precision Life. I'm going to ask him to say a few words about himself, his background in the health industry, and what Precision Life does. Steve, tell me a bit about yourself.
Thanks very much, David. Yeah, I'm Steve Gardner. I'm CEO and founder of Precision Life. I have been in the life science and healthcare space for 20 odd years. I started off as a structural bioinformatician, working on protein modeling, and quickly moved over to run research informatics for Astra, the pharma company. I finished that back in just before the Human Genome Project really kicked off. And since then, I've been involved in building innovative solutions that are really designed to make more effective use of information in deciding, which drugs to develop, which drugs are going to be safe, which drugs are going to work for which patients. And that's broadly speaking what we do today.
Okay, so were you involved in the Human Genome Project in any way, or?
Yeah, I was. We had an organisation at the time that was collaborating both with the academic and the commercial side of the Human Genome Project, and we ended up building information integration platforms that were used by companies, for example, like Insight Genomics, and they were using that to correlate information from not just from their genome-wide sequencing efforts, but also to add a lot of contextual information from other different sources. So proteomics was just starting in those days. We had the very early transcriptomics, and then there were a whole variety of other cellular assays and various other platforms. We basically acted as an aggregation center to pull all of those data together, some of the very first semantic integration platforms.
It's all very complicated, even for a techie like myself. But I remember I was very impressed when we had our last meeting by some of the things that you were able to discover about cross-purposing of drugs and understanding problems with multiple conditions and different drug mixtures. Do you want to say a few words about that?
Yeah, so what we do at Precision Life is we got set up, broadly speaking, to fill the gap between this increasing ability that we have to collect data about individual patients. So obviously the Human Genome Project is a big part of that, but we also have epigenetics, we have high quality imaging, we have epidemiological data, and of course electronic medical records now. The problem is, while we're very good at collecting it, we're not that good at always at using it, particularly, if you imagine in the context of a doctor, figuring out what all of that means in terms of which compounds are likely to be most effective for patients. And so we set up Precision Life as an analytics platform and company, really to start drilling into that. Now, there's a limit to where existing technologies can get you. If you take a genome-wide association study, which is the key tool that's used to go from large patient data sets to understand which genes are involved in that disease, the problem is they only work one mutation, one SNP, one gene at a time. And very few of the diseases that we're studying are monogenic. You have sickle cell and cystic fibrosis and Huntington's that are, but dementia, cancer, cardiovascular disease, they're all multifactorial diseases. And so the key difference that we have is that we analyze for combinations of features, and they can be combinations of mutations, plus epidemiological and phenotypic clinical data. we find those combinations that are really associated with the disease form that a particular patient subgroup has. And if you can do that, if you get that really high resolution view, it allows you to compare and contrast what's driving disease for two different patient subgroups. And right there, you have the opportunity to understand what's common and therefore likely to drive disease and what's different. And in other words, how to get precision medicine, how to tailor a therapy to an individual patient.
Yeah.
So along the way, one of the things that we do is build a really detailed architecture of complex diseases. So we cluster patient subgroups and all of the features that are driving their form of disease. And that allows us to go in and identify where we could find new drug therapies. But it also lets us bring in all of the existing formulary, all the existing approved drugs. Understanding their targets, we can map that back to genes, we can map that back to mutations, and then basically overlay them on this architecture of disease. So we can do systematic repurposing once we understand what's driving disease within the patient subgroups.
So when we come now to COVID-19, what kind of work are you doing in this area?
So we're working with some of the largest organizations that are collecting patient data, and we're looking to do a number of different things. We're obviously looking to understand the differences in host response. Why do people have different severities? Why do they have different pathologies? We want to understand that both at a genetic or genomic level, but we also realize that there's a whole bunch of other features. So there's smoking, there's gender and ethnicity, there's blood groups, underlying health issues or comorbidities. And understanding how they all play together is really important. That will help us develop tests that can be used to stratify high and low risk people. We can also do the systematic repurposing that we discussed earlier. And of course, we can also look for novel targets, particularly in that late stage of disease, where it's not the virus that's causing the real problems, it's the immunogenic inflammatory responses of the patient that's causing problems.
Okay, I think this is going to be extremely important going forward, and I thank you very much for sharing that with us, and also for agreeing to be a panelist on this upcoming virtual conference.